Now let us study the exogenous pathway of antigen processing and presentation, or MSC class 2 pathway. So the source of antigen in case of an exogenous pathway is always extracellular, or generally extracellular. The extracellular proteins can be internalized by means of endosomes by the specialized antigen presenting cells. So different types of antigen presenting cells have different binding affinity and efficiency to bind to different antigens. So generally, an antigen presenting cells have receptors that bind to the antigen patterns, like the macrophages express receptors for Fc proteins of antibodies and complement proteins of C3b. So they can bound to the antigens which are bounded to antibodies or the complement proteins. The surface immunoglobulin on the B cells also has high affinity and efficacy to internalize proteins which are specific to that immunoglobulin. The first step in an exogenous pathway is the endocytosis of the protein, and the second step is digestion of the internalized proteins. So this digestion generally localized to endosomes, which is in communication with lysosomes. So the late endosomes are rich in MSC class 2 or compartment, or MSC class 2 compartment MIIC, which is a key player in antigen processing and presentation. So however, some of the microbes such as Mycobacterium and Leishmania may survive and even replicate in the phagosomes or endosomes and persist in there. Similarly, autophagocytomes may also result in display of certain cellular proteins which are displayed on MHC2. Viral antigens specific to CD4 T cells may also be displayed through autophagocytosis. The third step in exogenous pathway or class 2 pathway is the biosynthesis and transport of MHC2 molecules to endosomes. These MSC2 molecules are prepared or synthesized in the endoplasmic reticulum, and both the alpha and beta chain are synthesized and folded by a protein called calnexin. However, these chains have this cleft, which is the one to which the peptides bond, is basically bounded to an invariant chain or called I chain, which promotes folding and assembly of MHC2 to late endosomes and lysosome. These MSC2 molecules are then transported via exocytic vesicles, and then they fuse with the endocytic vesicles of the antigen. Another protein called cathepsin acts on the invariant chain and cleaves it. It leaves a remnant called class 2 associated invariant chain or clip. Then another protein called HLA-DM facilitate the removal of clip and result in addition of peptides to MSC2 cleft. So when MSC2 is bounded to the peptide, it becomes stabilizes, and then it is displayed on the surface of antigen presenting cells. So these MSC2 molecules are recognized by T helper cells or CD4 cells. In this figure, a summary of class 2 MHC pathway of antigen presentation is shown. The first step is the uptake of the extracellular protein into the vesicular compartment of the antigen presenting cell. So these endosomes then fuses with lysosome as a second step, and the antigenic protein is then degraded into peptides. So the second step is the synthesis of synthesis and assembly of class 2 molecule, which takes place on the endoplasmic reticulum. And this alpha and beta chains are then held together by an invariant chain which bounds to the cleft of the MSC class 2 molecules. These molecules are then transported to Golgi bodies, from where they are released via exocytic vesicle. This exocytic vesicle carrying the MSC class 2 molecule then fuses with the lysosome, and within this cavity the invariant chain is removed and antigen is loaded onto the cleft of MHC class 2 molecule. So then the class 2 molecule carrying the antigenic peptide are displayed on the surface of the antigen presenting cell, and they correspond or interact with the T helper cell or CD4 cells and result in their activation. Now let us see the function of class 2 MHC associated invariant chain and HLADM. Basically, the invariant chain, the II, promotes folding and assembly of MHC class 2 to late endosomes and lysosomes. So this invariant chain remain bounded to the cleft of the MHC class 2 molecule. This invariant chain is then cleaved by a protein called cathepsin, leaving behind a remnant of 24 amino acids called the CLIP, or class 2 associated invariant chain peptide. This invariant chain is still bounded to the cleft to which the peptide has to be bounded. So this has to be removed. This removal is facilitated by HLA-DM, which catalyzes the removal of cleft, and now the cleft is free, to which then the peptide antigen bounds, and this bounded MHC2 class molecule are then exocytosed and displayed on the surface of the APC to interact with with T helper cells. Protein antigen, which is foreign in origin, but it is synthesized within the cell. Like in case of if we infect a cell uh, with a gene which contains ovalbumin, so this gene would be then then transcribed and translated within the cell, and this would be a foreign protein antigen which has originated within the cell. So this antigen would be endogenous in nature and would result in display of MSC class 1 molecule carrying the peptide of the endogenous antigens. So these peptides will only be recognized by CD8 T cells or cytotoxic C cells. So because they are displayed by class 1 molecule. So cytotoxic T cells would be activated and T helper cells will not be activated. So this is called CD8 MHC restriction. So in second case, you can see when we introduce an artificial foreign antigen directly into the cytoplasm. For example, we give an osmotic shock to a cell, and it results in uptake of ovalbumin, which is a protein, into the cell. So this is a direct uptake of a protein into the cytoplasm, and this protein within the cytoplasm be digested and processed and would be displayed by class 1 molecule. Again, only CD8 cell or cytotoxic T cells will be activated, and T helper cell will not be activated, as only MSC class 1 molecule are displayed, so which can only interact with cytotoxic T cells. In the third case, you can see there are if there is endocytosis of extracellular foreign protein, then these proteins are basically for extracellular in origin, so they will be taken up by the antigen presenting cell and will be displayed, and the protein will be degraded and will be displayed by class 2 molecule, as class 2 molecule only interact with the CD4 T cells or the T helper C cells. So in this case, T cell response will be T cell or T helper effector response will be generated, and cytotoxic T cell will not be activated. Now let us compare the features of class 1 and class 2 MHC pathway of antigen processing and presentation. So a composition of the stable MHC complex. In case of class 1 pathway, it is composed of alpha chain, beta 2 microglobulin, and the cleft is formed by alpha 1 domain and alpha 2 domain. In case of MHC2 pathway, the complex is formed between uh, alpha and beta chain and the peptide. So class 1 MHC pathway can be taken up by any nucleated cells which can act as antigen presenting cell. However, class 2 pathway can only be carried out by dendritic cells, mononuclear phagocytes, B lymphocytes, endothelial cell, and thyme epithelium. So this pathway results, MHC class 1 pathway results in activation or of CD8 or cytotoxic T cells. However, MHC2 class A pathway results in activation of T helper cells or CD4 T cells. The source of protein in MHC class 1 pathway are cytosolic. In some cases, they can be extracellular as well as the one which have escaped the phagosomes. In case of MHC2 class pathway, the source of protein can be endosomal or lysosomal. So mostly they are extracellular uh, proteins. So cytosolic proteasomes are the enzyme which are responsible for peptide loading onto MHC1 molecule in class 1 pathway, whereas in case of class 2 pathway, endosomal and lysosomal proteases are involved in binding of MHC to the peptide, such as cathepsins. So site of loading in case of MHC class 1 pathways endoplasmic reticulum, and the site of loading of peptides to the MHC molecule cleft is the vesicular compartment in class 2 molecules. So molecules which are involved in transport of peptides and loading include chaperones and TAP in ER in case of MHC1 pathway. In case of MHC2 pathway, chaperones of ERs as well as invariant chain and Golgi bodies and are involved in the transport of peptide and loading of MHC molecule. Third type of pathway by which an antigen can be presented and displayed by a MHC molecule is called cross presentation. In cross presentation, the antigen is basically extracellular in origin, but it is released directly into the cytosols, so therefore they are displayed by MSC class 1 molecule. An example is the case in which dendritic cell captures a virus infected cells or tumor cells, and these endosomal vesicles are then transported to cytosol by a unique traffic known to dendritic cells. So these peptides generated from the viral antigen can be displayed by MSC1 molecule, which then activates cytotoxic T cells. So during cross presentation, the same peptides can also be displayed by the MSC class 2 molecule, which are then recognized by CD4 T helper cells. So in cross presentation, both the MSC1 and 2 molecules can be activated and carry the peptide and results in the function of both T helper and T cytotoxic T cells. So up till now we have studied how the proteinaceous antigens are presented to T cells. So how are non-protein antigen presented? Let us study that. So there are several small population of T cells that are able to recognize non-protein antigens. These include natural killer T cells and gamma delta T cells. Natural killer T cells are the one which expresses both natural killer markers and T lymphocytes marker on their surface. So they have T cell receptors made up of alpha and beta chain, but they have limited diversity. So these NK cells, NKT cells recognizes lipids and glycolipids displayed by MHC-like non-classical molecule called CD1. So CD1 are the molecule which display the non-protein antigen to the NKT cells. So these NKT cells then recognize the lipid antigens and play an important role in defense against microbes which are rich in lipids such as mycobacterium. The second type of T cells which are involved in recognition of non-protein antigen are called gamma delta T cells. So these T cells can recognize some proteins, lipids, some phosphorylated molecules, and alkyl amines. As these molecules, non-protein antigens, are not displayed by MHC, so there is no MHC restriction in them.